Hello everyone. Today we are going to explore one of the biggest milestones in biology, the Human Genome Project. This project was a turning point in understanding what makes us us. As you have learned, the sequence of DNA bases contains the genetic instructions of life. If two individuals are different, that's because their DNA sequence differs. But what exactly is the full DNA sequence of human? In 1990, scientists launched the ambitious Human Genome Project, a 13-year international mega-project aimed at decoding the entire DNA sequence of a human, all 3 billion base pairs. At the start, it was estimated to cost about $9 billion $3 for every base pair. To store this information, there is need of 3,300 books with 1,000 pages each. The project generated an enormous amount of data. It needed high-speed computers and gave birth to a new science, bioinformatics, which deals with storage and analysis of biological data. Here are the main goals of Human Genome Project. It identified 20,000 to 25,000 human genes, sequence all 3 billion DNA base pairs, store this information in database, develop better tools for analysis, share technologies and industries, address ethical, legal and social issues known as ELSI. The project was led by the US Department of Energy and NIH, with key contribution from UK, Japan, Germany, China, France, and more. It was completed in 2003. Let's talk about how they did it. Two main approaches were used. EST method. In this method, sequencing only expressed the genes, and the sequence annotation. In this method, sequencing the whole genome blindly and assigning functions later. DNA was broken into small fragments, cloned into hosts like bacteria or yeast, using vectors like BAC and YAC. Sequencing used an automated methods based on Frederick Sanger's technique. The sequence was aligned using powerful computer algorithms. Only in 2006, the last chromosome 1, was fully sequenced. Here are some fascinating findings from the Human Genome Project. Total base pairs are 3.16 billion. Average genes 3,000 bases. Largest is dystrophin, 2.4 million bases. Estimated 30,000 genes, fewer than expected, and 99.9% of bases are the same in all humans, and less than 2% code for protein. Huge amount of repetitive DNA present. The chromosome 1 has the most genes, while chromosome Y has the fewest. About 1.4 million SNPs, single nucleotide polymorphism, were discovered. What does the future look like after Human Genome Project? The sequence opened doors to better understanding of disease, personalized medicine, agricultural and environmental solutions, studying entire gene networks and system. From studying just one gene, we now analyze entire genome. The Human Genome Project has empowered research across biology, medicines, agriculture, and more. Let's recap. Human Genome Project decoded 3 billion DNA base pairs, found around 30,000 human genes. Most DNA is non-coding or repetitive, enabled rise of bioinformatics and genomic medicines, contributed globally by many countries, completed the project in 2003. The Human Genome Project was not just about sequencing, it was about understanding life at its most fundamental level. This knowledge will continue shaping science, medicines, and technology for decades to come. Did you know that 99.9% of DNA sequence is the same in all humans? But what about the tiny 0.1% with a genome of 3 million base pairs? Even 0.1% difference means around 3 million base difference. These differences make each of us unique. These small variations are responsible for your phenotypic traits like height, skin tone, eye color, and even disease resistance. But to compare all 3 billion base pairs every time is impractical and expensive. So, how do we compare individuals quickly? The answer lies in a clever shortcut: DNA fingerprinting. It does not check the whole DNA but focused on special areas called repetitive DNA. These are regions where a short DNA sequence is repeated many times. When DNA is spun at high speed during density gradient centrifugation, the repetitive DNA separates out, called satellite DNA, based on sequence, size, and composition. They are grouped as microsatellites, mini satellites, etc. Let's understand why these sequences are useful. They don't code for proteins, but they show high polymorphism, meaning they vary a lot between individuals. They are inheritable, making them perfect for identification and paternity testing. Since DNA from blood, hair, skin, saliva, or even bones contains the same polymorphic regions, DNA fingerprinting works for forensic cases and inheritance studies. Now let's simplify the term DNA polymorphism. It means genetic variations caused by mutations. If a mutation appears in a germ cell and spreads in a population and occurs in over 1% of the population, it is called a DNA polymorphism. Since most of these polymorphisms are in non-coding regions, they build up across generations without affecting survival, making them useful for tracking diversity and evolution. The technique of DNA fingerprinting was developed by Alex Jeffrey. He used regions called VNTRs, variable number of tandem repeats, a type of mini satellite DNA, which show high polymorphism. Let's go through the steps of original DNA fingerprinting method. First, isolate DNA from the sample. Second, cut the DNA using restriction enzymes. Third, separate fragments by gel electrophoresis. Fourth, transfer fragments to a nylon or nitrocellulose membrane. Fifth, hybridize the radioactive VNTR probes. Sixth, detect band using autoradiography. Each individual's patterns of VNTR is unique, just like a barcode. Only identical twins share the same fingerprints. Band patterns differ from person to person due to different number of repeats. Thanks to PCR polymerase chain reaction, we now need only one cell to do DNA fingerprinting. It's fast, more sensitive, and does not need radioactive probes anymore. Applications help in solving criminal cases, paternity testing, studying genetic diversity in the population, tracing evolutionary relationship based on repetitive DNA like VNTR show polymorphism variations between individuals, inherited from parents, used in forensic paternity and population genetics, developed by Alex Jeffrey, now enhanced by PCR technology. So, students, DNA fingerprinting is not just science, it is identity at a molecular level. From the crime lab to evolutionary research, it is one of the greatest tools we have